Hello, everyone. Welcome and thank you for your attending to our third sessions of the EMN live webinar series. My name is Kim Boon. I am currently a scientific evaluator in National Pharmaceutical Regulatory Agency, Malaysia. And on behalf of the Early Member Network Committee of the Metabolomics Society, I will be co-hosting this event with Milena Barros Santos, a postdoc researcher at Bordeaux's Metabol Hub in France. Thank you for joining us. We encourage you to connect with us online via Facebook and Twitter for future content. And feel free to email us with suggestions or input. Today, we have two talks titled Multi-omics Data Integration to Reveal the Metabolomics Dark Matter.
Thank you for being here today. I am Milena Barros Santos, and I will introduce the first talk from Dr. Tiago Leão at the State University of São Paulo in Brazil. Dr. Tiago Leão is originally from the north of Brazil, from a humid and hot city named Belém, close to the delta of the Amazon River and surrounded by the Bay of Guajará. In 2014, he received his bachelor's degree in biotechnology at the Federal University of Pará, primarily volunteering at the Schneider Lab, where he started to learn about genomics, phylogenetics, and molecular biology. He performed 1 year of undergraduate studies as an exchange student at the University of Michigan, supported by the Brazil Scientific Mobility Fellowship from the CAPES Foundation Government of Brazil. At the University of Michigan, he spent a summer volunteering at the Sherman Lab, where he became interested in natural product research. Back home, he decided to apply for PhD programs to use genomics and molecular biology to search for new natural products. He obtained his PhD in marine biology at the University of California, San Diego, once again, fully supported by the Brazil Scientific Mobility Program. At the Gurevich Lab, Scripps Institution of Oceanography, he developed new pipelines and tools for understanding and exploring the natural products' rich strands from the cyanobacterial genus Morea. Next, as a postdoc in Dorriston Lab, Skype's School of Pharmacy and Pharmaceutical Sciences, he developed a tool for connecting mass spectrometry fragmentation data to their corresponding biosynthetic gene clusters. Afterward, he returned to Brazil and spent a year as a postdoc at the Fiore Lab at the University of São Paulo, further developing this machine learning tool, Multi-omics, and applying it to underexplored Brazilian cyanobacteria in the search for the new natural products. He is currently a postdoc at the State University of São Paulo, UNESP, in the lab from Professor Ian Castro Gamboa, working on metabolomics and machine learning tools.
Our second talk is from Professor Dr. Alexey Gurevich at the Saarland University in Germany. He will continue to talk multi-omics data integration to reveal the metabolomics dark matter. Professor Dr. Alexey studies software engineering at St. Petersburg State Polytechnic University with Bachelor of Science in 2010 and St. Petersburg Academic University of the Russian Academy of Sciences with Master of Science in 2012. In 2011, in the Professor Pavel Prafsner's group at the Academic University, he started focusing on algorithmic bioinformatics. The group later relocated to St. Petersburg State University, where Alexey obtained his PhD in bioinformatics in 2018. He worked at St. Petersburg University until 2022 with short research visit to the Prof's lab at the University of California, San Diego in 2015 and 2016, and the co-bachelor lab at the University of Tübingen in 2019 with DAAD fellowship. Since July 2022, Professor Alexey has led an independent research group at the Helmholtz Institute for Pharmaceutical Research Saarland HIPS and held a junior professor position at Saarland University, Germany. Professor Dr. Alexey Gurevich's research focuses on creating efficient algorithms and software for handling noisy omics data. He contributed to the state-of-the-art SPADE and QUAS genome assembly and analysis software and multiple computational methods for natural product discovery, such as D-replicator, VAR-QUAS, and NERPA. Most of his metabolomics tools are available as online workflows on the GMPS platform. Dear participants, remember to submit questions throughout the presentation in the chat or the Q&A box, and include who the question is directed towards by indicating the speaker's first name followed by the questions. I will give the presenter control to Dr. Tiago Leão, and the platform is now yours to begin presenting.
Hello, everyone. Thank you for being here today. Thank you very much for the invitation to show a little bit about our work on multi-omics and how to explore the biosynthetic dark matter. Today, we're going to especially talk about 3 tools. NPomics is the first one we're going to start. Let me share my screen so you can follow me. And then Professor Alexey is going to talk about NERPA and RP minor. So I'm just going to start. Thank you again for the introduction as well, Milena and Kim. I'm just going to start giving a brief background about the talk, about multi-omics, and also about myself. So like it was mentioned, I started doing undergrad more in research related to genetics, doing some chromatograms of genes, so a small portion of genes. And then I moved into more chemistry-based research, so started to connect the chemistry with the genetics. And then right now we are developing a more bioinformatic focused research on connecting again chemistry with genetics, and we developed the tool called NPomics. I just want to mention that I think it's highly important for bioinformaticians to know a little bit of programming. So we're gonna start an online free class in programming in Python and how to use bioinformatics and chemoinformatics. So if you wanna stay tuned for that, there's the NPomics website. 1 second. So I'll post in the chat the NPomics website, and then there you can find a little bit more information about NPomics itself. Also, you can submit your samples online, and then we can run them for you. But there is also some workshops on how to run the samples yourself. And then here is going to be posted about the class on Python as well. If you want to run NPomics, I suggest checking out the GitHub repository. I'll also post in the chat in a second the link, because there is some instructions on how to install it and how to run other tools that are required to run NPomics. And then there is a very nice quick 15 minutes video explaining the whole process. Okay. I'll post the links in the chat while the next presentation is going on. So let me move ahead and with the introduction. I just want to start. I mean, that this audience knows exactly the importance of natural products for drug discovery, but it's always nice to cite that a lot of the drugs are either naturally occurring or inspired by natural products, especially antibiotics. So how are those natural products made? It's important to highlight, especially because we're talking about multi-omics, that it's encoding the DNA, which it's called the sometimes it's called the code of life because it has all the information required to make the cell in the DNA. Then that information that gets transcribed into RNA and translated into proteins. And normally that's the code of life that we learn on high school. But we know from the natural product background that some enzymes, some kinds of proteins, they have the domains that put together scaffolds. So small molecules, also known as natural products or metabolites, secondary metabolites. So then there's an extra layer, which is the enzymes putting together the metabolite. In general, general pipeline for drug discovery includes sampling, culturing. It can be skipped. You can do microbiome samples. You can do full metagenomic samples from nature. But you can also culture to enhance a little bit the signals you're looking for. And then normally it's a bioassay guided isolation. So we tested some kind of activity. It could be antimicrobial, antiviral, anti-cancer. There are many different kinds of activity, and that's one of the challenges to know what kind you want to test. But once you find a hit, something that is active against the queried activity, then this crude extract, a mixture of metabolites, can go through the isolation process, which is normally chromatographies to separate the metabolites into like pure compounds, and then you need to retest the bioactivity, and then you go to the NMR, nuclear magnetic resonance structure elucidation. And then if you want to really develop as a drug, then you need to know also the mechanism of action. So there are several steps, and then there are several limitations in each step. For example, not everything can be cultured in the lab currently. Again, which activity you need to test, and it's a very repetitive process, the bioactivity guided isolation. NMR structure elucidation can be quite challenging sometimes. It can take months and sometimes even several years. And also mechanisms of action is pretty challenging. So coming from a mass spectrometry perspective, so we do mass spectrometry and then normally couple before with chromatography, and then we have mass spectra as our output. So now the tool has to parse that output from the mass spec and then come up with predictions. So what kind of predictions can you do? For example, there are predictions of novelty and how diverse is the new metabolite. There are predictions of bioactivity and structure. However, if you're only using a mass spectra, then if you only have one mass spectrum and you cannot replicate, you cannot match to anything in the library, so it's really hard to predict the novelty because first, not everything is expressed by the bacteria, the fungi, the plant. So there's that certainly some things that are missed at the snapshot that we take on the chemistry at that given moment that we extract the sample. Bioactivity tools are lacking for predicting bioactivity from spectra. And also structures are difficult, and stereochemistry is actually not possible to predict just using mass spectrometry. So that's what we call then the metabolomics dark matter. We can see the spectra, we can see a particular spectrum that is interesting, but we cannot predict a lot of the things that we wanted to predict. And then that's where he enters multi-omics. Multi-omics is able to make some of those predictions and is evolving quite quickly on how to make those predictions. So start talking about NP-omics. I'm gonna give a brief overview. So this is the publication, it came out last year. So NP-omics is really a tool that is, the main objective is to assign genes to unknown and known mass spectra, so mass fragmentation data. So we're trying to connect the gene clusters. They're called clusters because they're physically grouped together in bacteria. Right now, NSMASH is only for bacteria. You can try other organisms, but the success rate is probably going to be very low because the database is it's only bacteria right now. But we're working on a second version for plants and fungi, and then we're gonna expand for other organisms in the future as well. And it's a machine learning tool, so you give some data to the machine, it learns the patterns in the data, and then it tries to make the prediction, which is what gene cluster is making this new metabolite. So just quite, I'm gonna just brush through it, but we have genomic and metabolomic pair data. So the same sample you collected and made a genome, and then you collected the LCMS data and made a metabolome, so several metabolomes and several genomes. And then you can use the database, the paired omics database, to get more samples, because the more samples you have, the better is the learning process. Then there's a pre-processing step, which is networking. So we use in the gene side, the Bigscape tool to network samples. And in the metabolomic sides, we use GNPS. And then once we have the networks, we can parse them into tables. And that's the input for the tool to learn and predict gene clusters for new metabolites. And then you can use other tools, like was mentioned, Professor Alexey participated of the development of the replicator. So that's a pretty nice tool as well that can be combined. MassQL is another new tool that can be combined as well. So it's a very powerful approach to combine multiple multi-omics tools. And this is the kind of output you get from NP-omics. So it's a predicted biosynthesis. So you have every domain that the genes are encoding, and then every step that you predict that is happening to the metabolite. In red, we have a slightly more difficult steps to predict, because those are hydroxy acids instead of regular amino acids. But then at the end, we get a structure. It could be a partial structure. It could be, in this case, a full structure. So that's able to, you can determine how good is the structure by the difference between the delta mass between the observed versus the predicted. And this is a real this is a real scenario as well, but this was the second one here was manually made. So you can see that we get a very similar kind of output that it can be done manually, but the time and effort to do it manually is much larger than the machine doing it automatically. And then, so this kind of table, it's the core of the machine is the data that is gonna find the patterns on it. It has similarity fingerprints. So again, we do the networking, so we get scores of how similar things are, and that's part of the fingerprint that the machine is gonna learn. Fingerprint is just basically a group of values that teach the machine about the data. Then we can also put the class like terpenes, polyketides, peptides, and so on. And we can put some predictions about what can be in the structure. So for example, here, tyrosine, proline, amine, and malonyl-CoA from PKS, and then so on. So once you can do those different kind of fingerprints, you can test the tool. And then we tested several different tools together. Some tools in pink here we were not able to test because they're discontinued. And then you can see that NP-omics had a pretty good performance. It was pretty precise, and it was able to analyze a good portion of the data. In the case of using substructure and similarity and using a cutoff, we were able to analyze the whole data. And then 77% of the answers were correct. This is a very small validation set, so we need to perform more through validation. But this is a good indication that NP-omics is working quite properly. Okay, so the last portion of this first part of the talk, and then Professor Alexey will continue, is some outputs as an example. So the validation, for example, there are several metabolites that were able to connect properly with the the gene clusters. So this is the kind of output. Here's the metabolite identification, and then here's the prediction. So we predict that those are the, oh, sorry, this is the true answer of the gene that are making those metabolites. And then in this case, we can ask how many predictions NP-omics is going to give us. The more predictions, the higher is the chance of one of them being correct. And then normally, very commonly, only one prediction is correct. It's very rare for two gene clusters making one metabolite. It's more common for one gene cluster to make several metabolites. So here in black, you can see which predictions are the correct ones, and only one prediction was not assigned properly. So let's grab the example of albocytin, and then here's another way to look at the output. We can look at since it's a known metabolite, we can look at the structure. We can see where it's gonna fragment, and we can match the connected fragmentation data with the gene cluster. And then you can see here's just a plot of the fingerprint. So it's only present in one strain out of 1,040. And then the metabolites are present in the same strain. So that's why they are correlated, besides being the same class as well. You can also visualize the outputs in the network. So for example, you can use Cytoscape and then see here that the gene cluster for rosamycin is connected to the ions, also annotated as rosamycin. Same for selenamide and the spherioctamines, but it's not true for actinomycin. All the genes that were connected were false positives in this particular case. So the two is not perfect. It's the prediction too. It can make false positives as well. So it's good to have the research background to go and look and see which ones are more likely true positives. For example, you can do that looking at how many members are in each class. So for example, how many members are in the hectachlorine family, which is green, which has more members than the Jamaicamine family. So the squares are ions, and then the circles are the genes. So you can see that the abundance of the genes, so how many members are in the gene family, it's matching the Jamaicamine metabolite family. So that's an indication that this is probably the correct answer. Also, we know the answer is Jamaicamide, so we know that this is correct, and not hectachlorine, which is a different metabolite. And we can see here by the colors that the size of the family are distinguished. So yeah, so these are the correct ones, and then we have some false positives as well. And you can do that for unknown metabolites. So in here you can see that the darker blue is or green, greenish. It's the one that has the most number of family members, and then it matches this other one here, even though we don't know exactly what are those metabolites. We just know that this is a peptidic one, and then we have some hits that are steroids, and then this one is not particularly classified. So the last thing I want to talk about is that we can also make structure predictions using the genomic data. For example, once NP-omics output something, a link between the metabolite and the gene, then we can make predictions of the structure, and you can see the difference between the predictions we can make using the genes. So supervised BGC, biosynthetic gene cluster prediction, we can predict a lot of different things. And then from the MS side, we can predict what's missing, and then we can calculate the error. So that's another option that you get now with NP-omics. You can get those outputs and then make those predictions. And then with the metabolite side, we were only able to make one predictions because the other amino acids are modified, so they're a little bit harder to predict. So yeah, there are several limitations. There's no ideal number of examples per class, so the classification needs more examples. We only run one iteration, so there's no reinforcement of like teaching the machine how to learn better and better. It only learns one time. There's no molecular family information, so how the molecules are connected. You see here, they're not connected between each other yet, but we're working on that. The validation set is a little bit small, so it's only 89 ions, but again, we are also improving that. And then we're gonna use also combination of other tools, like Professor Alexey now is gonna explain about NERPA and then Prominer in a minute. So we can combine that with NP-omics and make those kind of visualizations using multiple tools. And then the last thing is that batch effects and co-occurrences can be also a very big issue, because it can be that a contaminant just happened to be present where the gene is present, and then you have a false positive. So thank you very much. I have a lot of people to thank about this first part of the talk. Now Professor Alexey is going to continue with the talking about NERPA and NRP minor. So a little bit more about peptidic genome mining.
Hello, everyone. Let me share my screen. Okay. Okay, thank you for the invitation and for the kind introduction. And I will continue the talk by Tiago, but my talk will be a bit more specific, not about metabolites in general, but about specific class of metabolite, which called non-ribosomal peptides. And these compounds are small and usually produced by microorganisms, but they are quite important, and many medicines are in particular non-ribosomal peptides. And you probably know penicillin is also a promising and notable example of them, but not the only antibiotic and not only a natural product drug, which is non-ribosomal peptide. And they are synthesized by specialized enzymes called NRP synthetases. And these enzymes are encoded in genome, usually not in a single gene, but in large biosynthetic gene clusters. I will use abbreviation BGC, which is quite common in the field. And these BGCs may span dozens of kilobase pairs. So if you consider bacterial genome as a few millions of kilobase pairs, a large portion of it could be devoted to produce just a small NRP, which highlights their importance. And core genes responsible for NRP synthesis are in turn organized in models, and each model adds one additional amino acid to the final NRP product. And model in turn consists of several domains. Some domains are always present, some are optional, and are responsible for modifications of these amino acids. We can consider NRP synthesis as a large assembly line, like car assembly, for example, and usually there is a single starting domain. Here it's colored blue, and several elongation domains. Here it's colored green, and one final termination model, which is yellow here. And common and required domains are A domain, which is responsible for selecting particular amino acid, which will be added to the final product. T domain or PCP domain, which connects carries this amino acid to the next model, and condensation domain responsible for organizing the peptide bond, and final termination domain, tRace domain, which releases the final product. And there are many, as I said, many optional domains which are responsible for modifications of these amino acids, which result in huge diversity of NRPs and their evolutionary adaptation to different conditions, which makes them quite bioactive. And here is a common example. It's surfactant A. That's how it's synthesized. It's NRPS is encoded in 4 core genes, and the final product looks rather small. And it's important to link NRP structure to their BGCs, because it can help in industrial synthesis of these products, because their producers are not always culturable bacteria. So it could be important to know exact cluster, which can then be heterologically expressed in other more friendly bacteria. And it will also can help to find new NRPs, which could be a promising candidates for antibiotics and other drugs. But this problem is challenging, and currently many NRPs are known. The largest database, bioinformatic database of known NRP structure is Norine database. It contains around 2,000 NRPs, but it's not the complete collection because they are widespread in other sources. And there are thousands or tens of thousands of NRP producing genomes already sequenced and available in RefSeq database, but only about several hundreds of NRP BGC pairs are really known and stored in the largest specialized database of BGCs, which called MeBig. So even for known NRPs, many BGCs are not discovered yet, and let alone for new NRPs. Why it's so? That's because there are many challenges on the way. First, it's not straightforward how to predict which amino acids will be added to NRP, the final NRP structure. Usually, these optional domains may drastically change amino acid. So even if you predict correctly, bioinformatically predicted, it could be different due to these optional domains. Many NRPs has non-linear structures, which is hard to deduce from the genome. And the last but not the least, synthesis is not always that simple as it's written in the genome. Some models and gens, even if present in bacteria, could be simply skipped during the synthesis or used several times, or order of gens activation could be not as in the genome. So there are many possibilities, and it's not straightforward to predict NRP from BGC or to predict the BGC from the final structure. And we developed a tool called NERPA, handling this, addressing these challenges. And it was published in late 2021. And I will not go into algorithmic details, of course. It's not the scope of this talk. I will just briefly show the pipeline of the tool and some of the results, and I encourage you to read the paper if you would like more details. So basically, our tool takes as input a set of non-ribosomal peptide structures in a common format and a set of genomes. We perform genome mining using ANSYS-MASH, which is a state-of-the-art tool for this problem, and as Tiago mentioned it. And for NRP structures, we try to perform retrobiosynthesis. That means to reconstruct which amino acids are inside the structure, because some amino acids may be modified post-translationally. And NERPA itself try to match these two types of data together to find the likely connected ones. And the outcome of our tool is a list of non-ribosomal peptides and corresponding BGCs in genomes with some scores. The higher score, the better. So key features of our tool is that we take into account reliability of a domain-based predictions. So prediction of amino acids is usually it's a bioinformatic or machine learning tool behind this, and it could be wrong, but we consider this possibility. We use information from all optional domains. So if, for example, methylation domain is present, we consider that corresponding amino acid could be methylated or not. And we learn our function based on manually curated data set of known pairs from the MBIG database. So our tool is rather accurate, and we try to support this tricky non-canonical synthesis features. And to test our tool, we use all representative bacterial genomes from RefSeq and our in-house database of putative NRP structures, which is about 8,000 of structures, but not all of them are really NRP, but it's usually not possible to figure out just based on structure. And there were many hits, and in the top 10, we found 6 already known BGCs and 4 potentially new ones. And we explored using mass spectrometry and manual analysis, one tentative novel BGC, and confirmed that NERPA was correct. It's a BGC for N-gerhiomycin. Here was the input for our tool. So this is N-gerhiomycin structure, which was known before, like for 15 years. And this is a genome of Photobacterium galatea, available also publicly online. We did genome mining and retrobiosynthesis of N-gerhiomycin to annotate it into amino acids. And then inside this BGC, there were 6 models responsible for 6 amino acids. And here are the predictions of specificity of these models, so which amino acids are likely attracted by these models. And numbers in parentheses are scores, so how likely these amino acids are there. And NERPA did the matching. Of course, this case is not that tricky, but remember we were searching of thousands of genomes against thousands of structures. So this match was among them. And here you see that there is perfect match for 5 predictions and 1 partial match where in real structures there is a leucine, and in predictions there was isoleucine, but NERPA can handle this. And later we found this example computationally and then ordered this particular bacteria, culture it, and performed mass spectrometry. And since N-gerhiomycin structure is known, it was easy to compute its mass and predict common and possible ions in mass spectrometry. And we observed mass spectra with all these ions there. And here is a network of similar mass spectra from this cultured bacteria dataset. And there are currently 9 known N-gerhiomycin variants, A, B, and so on. And you see all of them here in the network. So it's very likely that this particular BGC is producing this particular compound. And you see there are many unannotated nodes, which are probably new variants of this N-gerhiomycin. And as we know from other studies, some not very abundant variants of NRPs could be more clinically effective than their more abundant counterparts, which are already present in the databases. So finding new variants is quite helpful for medicine. And since here we have a webinar, I have a few tips how to run NERPA. So our tool is freely available and on GitHub, and we have a rather detailed manual how to run it with several use cases and examples, how you can inspect your genome or inspect your compound, again, many genomes, or do both. So you can go there and try the tool yourself on your data or on our test data, or write us if you have any issues. So in summary, for NERPA, it can efficiently screen a lot of genomes, against a lot of NRP structures in any direction. And there is only one alternative, and we outperform it in quality of results and computationally. So NERPA is quite fast, but the downsides that it currently has only a command line interface, which limiting its usage, especially in Metabolomics community, I suppose. And it doesn't have automatic integration with mass spectrometry data. So we did this manually in our case. And the last, and maybe the most important, it cannot discover completely novel NRPs. And the next tool, which I will present in a minute, overcoming all these limitations. But we also working on NERPA further in my group. So we hope to present web service for it maybe later this year. So stay tuned. And if you're not familiar with command line, hopefully we will provide you a more user-friendly interface and integration with mass spectrometry. But we already have another tool for similar problem. Again, it's for non-ribosomal peptides. It's called NRP Miner. And it was also out in 2021. And this tool is really multi-omics one. So it works with sequencing data and tandem mass spectrometry. And so there are two main stages in this tool. First is based on genome. So we do genome mining using anti-smash, and then try to predict all possible combinations of non-ribosomal peptides which can be synthesized by this particular BGC. As I told you before, there are many challenges on this way. So some domains could be skipped, some can be used several times. Amino acid specificity is not well predicted by current bioinformatic approaches, so we should consider a lot of possibilities. It's a large combinatorial problem, but an NRP miner try to use some heuristics and algorithms to make this set feasible. It's still large, but feasible. And in the second stage, we utilize mass spectrometry data to screen all these putative computational predictions in silico constructed non-ribosomal peptides against real mass spectrometry data to find only the most significant matches, peptide spectrum matches, PSMs, in this large data set, and then use them as a guide to expand this data set using spectral networks and do more exhaustive exploration using NMR or other sources. Again, I will not go into bioinformatic algorithmic details, just show you a quick overview of the results shown in this paper. We analyzed 4 bacterial microbial data sets of different origin, and you see there were many strains in the data, and we found using NRP miner many known NRPs from NRP families, because usually NRP, a single BGC produce not a single NRP, but a family of related NRPs. But also we found 4 new families and 25 new NRP structure, completely new, not similar to anything known before. And we did this automatically with just clicking a single button. And here is one of these 4 data sets. Each node on this figure is a spectra from this data set. And some are known. We annotate them using GNPS library. And colored nodes are significant hits of NRP miner. So every colored node is a spectra which was identified by NLP miner and linked to some NLP structure with high statistical significance. And you see, we found 3 new families with many statistically significant hits. And in the paper, we did analysis of these structures, and for some of these peptides, we elucidated the full chemical structure, did NMR and other measurements, and proved that this bioinformatic approach could be a nice tool in hands of experimental researchers. And again, you can use it. It's more friendly than NLP than NERPA. There is again well-documented README on GitHub where you can see step-by-step instructions how to run the tool, which is available on GNPS. So you will need 2 online resources: ANSYS for predicting for genome mining of your genomes, and GNPS for analyzing your mass spectra using NLP miner. And there is some instructions how to interpret results, and our test data also is there. And that's all for my side. And I would like to thank a lot of people involved in both of these projects. And if you have any suggestions or if you'd like to, so you are free to try the tools. And if you have any problems, just feel free to contact me or use official pages. And thank you for your attention.
Thank you, Professor Alexey Gurevich and Dr. Tiago Leão, for a great talk. There are questions in the chat. Even if they have already been answered by Tiago, it would be nice to explain verbally. Please keep them coming. The first one is for Tiago. You mentioned the NPO-mix platform is currently only available for bacterial omics, but plants and fungi are coming soon. What are the former missing to be complete and released?
Yes, so because it's a machine learning approach, we need a training data. So the training data needs, for example, one sample, let's say I have a bacterial culture or a fungi culture, and then for a plant that I just grabbed from the environment, I need to collect genomic data and metabolomic data at the same time. So I need to extract both DNA and metabolites. For the DNA, we run sequencing. It could be like Illumina platforms, and Nanopore is a very good one, PacBio as well. And then so you would need to assemble the data, which is kind of a little bit challenging as well, depending if you have like samples with more than one organism. So you can use microbiome data. You grab a gut microbiome and use the whole sample, collect genomic and metabolomic data. For the metabolomics, it's important to highlight it has to be mass spectrometry with fragmentation, so MS. So we cannot use just MS1 data. It does not require necessarily to run chromatography, but is also a very good step because this way you can acquire better spectra. So yeah, so the problem is that all of those requirements are only fulfilled mostly for bacterial samples in the database. So all the samples that are in the Paradomics, I'm not sure if new samples were included lately, but they were so far all bacterial samples. So if the Paradomic had more fungi plant samples, NPomics could be applied right away. So all we need is more people to contribute from the fungi community, plant community. So please, if you have genomic data and metabolic data in the same sample, pair data, please do contribute. I posted the link of the Paradomics in the answer, so you can contribute also with the database, and that would also help NPomics.
Thank you, Tiago, Dr. Tiago, For second question is also to you. The question is, what type of genomic information is required as input, and only genomic data or transcriptomic data is also useful for making predictions?
No, so yeah, you don't require transcriptome data right now. Transcriptome data, like the actual transcript value scores of like RT-PKMs, they're not supported at Multi-omics, but we are working also in the Multi-omics to not only to include plants and fungi, but also we're going to have new features, like it's going to be much faster, and it's going to include also transcriptomic data is going to be optional, because genomic data, so all we need is you need a FASTA file with your contigs, which are the pieces of the genome. So bioinformatician normally put together the genome data into contigs, and then NSMASH is the tool that you're going to need to use. I send also the link on the answer to process that in the genomic data. And then you can use Multi-omics. But the transcriptomic part is highly important to be included in the future, because right now, for example, one of the issues is that we have a lot of false positives. So one way to remove false positives is by removing genes that are not being expressed, so they're not making metabolites. So we can get that information with transcriptomics, but right now it's not supported on the current Multi-omics workflow.
Thank you, Tiago. The next question is for both. What type of information from genomics data is mostly useful to complement information we can acquire from metabolomics data?
Alexey, would you like to make a comment on this question?
Yeah, I can quickly comment. So usually from genomics, it could be important to know taxonomy of this genomics data, because as we saw recently in our experiments, prediction of a domain specificity, so NRP specificity, could be related to taxonomy of the samples. For example, if it's completely something from real unknown space, it could be hard to predict, and our tools should know this, that predictions will be not that reliable, or vice versa. If it's something very well studied like streptomyces or something like this, then we should rely on common predictions very well. So this information could be useful. What else? I saw you answered partially in chat.
Oh, just about the structures, I just want to compliment. So, for example, for peptidic structures, like we highlighted in the talk, Professor Alexey mentioned, it's a little bit more straightforward to predict. But we are also able to predict some parts of other kinds of structures, like some classes like terpenes or glycosylated terpenes. Also, some classes that they don't even actually they have their own class. They are just called others. So we were able to predict structures from those classes too. But normally it's quite challenging to predict structure. We are actually working together, me, Professor Alexey, a lot of other collaborators like Justin van der Hoff. Professor van der Hoff is also in the talk, present in the talk. We're working on a tool for structure prediction. So the idea is to use first NERPA, NRP minor, Multi-omics, all the multi-omics tools that we can make links between the metabolites and the genes, and then use that link to predict structures. So we're working on that, but it's definitely quite challenging. Machine learning can certainly help, but there's still a lot to work on that, especially for non-peptidic metabolites.
Thank you, Dr. Tiago and Professor Alexey. For the next questions, will the slide be shared? So for these webinar sessions, The recording will be uploaded in the Metabolomics web page. You may refer to the Metabolomics Society webpage later. And the last question to Dr. Tiago, a great talk, especially when you only have partial structures. How successful have you been in getting to the full structure of a target molecule? And is it solely relying on the fractionation process?
So we had 22 examples of different metabolites that we're using to test in Metabolomics. And then we, out of curiosity, it was not included in the Metabolomics paper, but we also tried to predict their structures without using library data, so without matching to the library. So as if the tool did not know, so the tool was not, we didn't give to the tool what structures they were, like this glutamaldamide, this Jamaicamide, this curacin. We did not mention, so the tool didn't know that. And it was able to predict only 2 full structures from 22 examples and 1 absolute structure. But then I wanna emphasize, this is not doing fractionation per se, because the sample is still a crude extract, or it could be a fraction, but it could be actually a crude extract. Well, the fractionation that does happen is you aliquot a piece of the sample, like tiny amount of milligrams of the sample, and then you run the LCMS equipment. So it's going to have a chromatography step that separates a little bit, and depending on your conditions, might separate more or less. But then, so the actual sample itself, it's still a mixture, a complex mixture. So we're able to predict full structures only using mass fragmentation data and genomics for mixtures. So we got 1 absolute structure without doing the actual isolation as our job, but it was previously done by other researchers, the isolation in NMR structure configuration. So it was confirmed the structure by NMR. So we had those examples that were validated, but the tool can predict the structure even without using isolation and NMR in 2 out of 22 cases.
Thank you, Dr. Tiago. I think we have addressed all of the questions in the Q&A box. So once again, we would like to thank Professor Alexey and Dr. Tiago for these sharing sessions. Dear participants and members, thank you for your time joining us for this fruitful event. We wish to see you again in our next webinar. Thank you and bye-bye.
Bye-bye.
Bye-bye.